Okay. So very warm greetings to everybody joining us in India, outside of India, different parts of the world. We are having our monthly webinar series as part of the Center of Excellence in Astrobiology at Amity University in Mumbai. And we are supported by Blue Marble Space Institute of Science. And for this webinar, we are proudly partnering with the Birbal Sahni Institute of Paleosciences in Lucknow, which is a Department of Science and Technology organization. And we have been closely working with BSIP Lucknow now for almost 5 years, 5 glorious years, in which we are establishing astrobiology research and building bridges to conduct more field work and lab work together. So today we are having a very special guest, one of the, you can say, prime mover and shaker of all our astrobiology efforts in India, Dr. Mukund Sharma, sir. And it is my privilege to introduce him to all the participants joining in, as well as those who are listening to us. So I will just read a little bit about Professor Sharma, who people are not aware of, would like to know. So Dr. Mukund Sharma, senior paleontologist and astrobiologist from Birbal Sahni Institute of Paleosciences in Lucknow, is renowned for his immense contribution in the field of Precambrian paleobiology in India. He has extensively worked on the Archaeans of the Dharwar Craton, the Proterozoic basins of peninsular India. He has reported synergetic fossil cyanobacteria from 2.6 billion years, Neo-Archean iron formation of Karnataka. His studies on 1.6 billion year old Vindhyan supergroup demonstrated that, I'm trying to read this name, Giraiphanes spiralensis, considered to be a eukaryotic fossil, is actually a coiled long sheath of prokaryotic cyanobacteria. So I hope sir will tell us more about these findings. And he has several different such similar discoveries and findings to his accolade. Dr. Sharma has co-authored monographs on Proterozoic fossil cyanobacteria and stromatolites of India and Russia. He has edited a special volume of Journal of Asian Earth Sciences on Proterozoic basin of India. Being a strong advocate of science outreach for general public, he frequently delivers popular science talks and also has written 2 popular books on fossil plants. His research contributions have been well appreciated by the national and international science fraternity. The Indian Science Congress Association had conferred on him the Young Scientist Award in 1992. Iyengar Sani and Sharda Chandra medals were bestowed upon him in 2009. He received the Chinese government fellowship in 1996, Boys Cast fellowship in 1997, IITP fellowship in 2000 and 2007, and Indo-Australian Senior Scientist Fellowship in 2013. Very recently, Professor Sharma received the prestigious L. Ramarao Award in 2018, which is given once in 3 years to the geologist who has made significant lifetime contribution to Indian stratigraphy and/or paleontology by the Geological Society of India. Amongst all of these very distinguished and prestigious awards, I would say that for the field of astrobiology research in India, Dr. Mukund Sharma's contributions are unparalleled. The amount of effort and work he puts in completely selflessly is unparalleled, and I'm a personal observer to this fact. So with this, Dr. Sharma, we are very fortunate to have you with us, and we would request you to take over from here, and we will have your lecture, and we will expect all the participants to write down their questions in the Q&A section, and we will try to take as many questions as possible. So please, Sharma sir, please take it away.
Okay, good morning, Siddharth, and thank you for the nice words you have mentioned. And good morning, good afternoon, good evening, and good night to all the astrobiology friends from different parts of the world. Am I audible to you, all of you?
Yes, sir.
Hello.
We can hear you.
Yeah, okay.
We can hear you. Yes, sir.
For the last 5 years, we are together moving towards making a center in India, and I'm very happy to see that Siddharth is able to do this in the western part of India, and he has opened a center of excellence in astrobiology with the help of Amity University. And we are very grateful to the Amity University also for giving this opportunity to a young man who can lead this particular center and bring all the laurels and the expertise to this center. I am very much confident. As Dr. Siddharth has mentioned that I have been working on the Archeans. So during this particular lecture, I will be telling you my experience in working on the Archean sediments of India, particularly the sedimentary successions from where what we have recorded, and in the other cases, how it can be used in the astrobiology, in the field of astrobiology, because we all are looking for the signature of life in different planetary bodies, mostly in the Venus and the Mars, and all of them are having almost the similar history as far as the development of lithosphere, atmosphere, and the hydrosphere, if it at all it was present there. And we are confident that the hydrosphere was also there, particularly in case of the Mars also. So I will be showing you what we understand at present about the Indian Archeans. And Archean is the one I'm sharing you now. Uh, So the title which I have chosen today is the Archean life and its relevance to astrobiology. As those who are coming from many people are here from different backgrounds. So I want to show that what we understand about the Archeans. In the Earth history, we have divided the time slices in by different names. And the Precambrian is one time span which holds the 5/8 part of this entire Earth history. If you see, our Earth is supposed to be 4,600 million year old, and the rest of the things which are coming on the Earth, they were on the in the Phanerozoic time span, which is just 541 million year old. So the rest of the time comes under the Precambrian, and Precambrian is broadly divided into three parts: the Hadean, of which we know very little about that. Then comes the Archean, which is start from 4,000 million years and going up to the 2,500 million year, and from 2,500 million year up to the 541 million year. That is the time period division between the Precambrian and the Phanerozoic. Phanerozoic is time period where entire evolution of the life forms and even the Earth has taken place. But the main body was taking place, means the advent and origin of life took place during the Archean. And if you see this particular chart, this is known as an international chronostratigraphic chart. It is revised frequently, and this is the third version of 2020, as you can see here. And there are several spikes are also put here. So these are the timelines which are very precisely defined. But if you see the Precambrian part, it is still not worked out. There is only one timeline, which is known as a golden spike. They are in the golden color here, where we have a definite timeline that we know that from this particular time zone, the other younger parts come to an existence, and this is known as an Ediacaran. Today in my lecture, I will not be covering this particular part, which is known as a Proterozoic. But since Siddharth had mentioned, that was the one part is the Grappinia, which comes in the topmost part of the Paleoproterozoic. So, but today I will be leaving that, and I will be concentrating myself towards the three divisions of the Archean, that is known as the Eoarchean, Paleoarchean, Mesoarchean, the Neo-Archeans. And here this is start from 4,000 million years and going up to the 2,500 million years. So you will have to keep that in mind, that whatever we are going to talk today is only concentrating on the Archean part. So the next slide. This is just to show you that how much time span is involved in the Precambrian. As I mentioned, that is starting from 4,600 million year and going up to the 541 million year. That is the time period that is known as the entire Archean, including the Hadean, Archean, and Proterozoic. When we come to the biosphere, because today I'll be talking more about the biosphere, because the Archean encompasses all the it comes under this scope is the lithosphere, hydrosphere, and biosphere also. But I will be talking more on the biosphere. And the Archean biosphere we can easily divide into the Stromatolite, microfossils, and megafossils. and then comes in the in the Proterozoic part, the metazoan remains. As I mentioned that we know very little about the head end, so we are not going to talk about the head end, but we are coming to the Archean, and even in the Archean we have a very little knowledge. But the oldest stromatolite, which was recorded some years back into 2016 by an Australian group, that is a 3,700 billion year or 3,700 million year old. And there was another discovery long back in 1983 that was again from the Australia, where from the Varavana group of Western Australia, stromatolite was also recorded. This is the domal shaped stromatolite recorded from 3,500 million year old rocks. So you see that these are the very faint evidences of stromatolite. This particular stromatolite is coming from Greenland area of the world. This is again, I am showing you, you see this is the entire rock piece, but the stromatolite are confined to this particular zone, or here they are, the laminated part are there. These laminated part, the domal parts, are made by the cyanobacteria. It is supposed to be in the, if you take a modern analog, then they are made by the cyanobacteria. But in case of the Archean, we consider that these were formed by cyanobacteria as well as some bacterial elements also, because in the earliest part of the earth or in the Archeans, we have a very little oxygen, as I will be showing you later in the lecture. You see that the different types of stromatolite which were recorded from the Varavuna group by Walter, Avramic, and Schoff. Walter was from Australia, and the Avramic and Schoff were from in America. So these are the laminated structures. These laminated structures suggest that biological activity was taking place. And even if today, if you go to some shore areas, you will find that there are fine laminites are there. Fine laminites are developing, but all of them do not convert into an stromatolite. Stromatolites are only formed when there is a calm, quiet environment for a long period, then only. And because nowadays all these shore areas are disturbed by the human activity, anthropogenic activity, as well as lots of animals are also there, which feed on these algal laminites. So they do not allow to form the stromatolites. That is why we do not get the stromatolite in the present day modern environment, except 2 or 3 places. One of them is in the Shark Bay in Australia. So how we find that there were biological activity was taking place. So if you take a rock samples, particularly the sedimentary rock, here I would like to emphasize that only the sedimentary rocks host the evidence of biological ability. And when you prepare a thin section, that is preparing a very fine thin section on the glass and mount on the glass slide and see under the microscope, then you will see some of the activities of the microbial activity. These are the microbial activity which are disturbed due to the long or very old age of the sediments. So this type of structures are recorded, but they are not many, they are very few. And whenever you get it, then you have to establish 3-4 things about that structure, whether what structure you are demonstrating and claiming it as a microbial activity, the Archean, then you will have to show that they were syngenetic, but that means that they were formed when the rock was depositing, and the age of the rock is also Archean in age. You have to demonstrate the syngenicity, and these structures are not abiogenic. So these are some of the very tough questions which you have to address, or anybody who is trying to understand the microbial life of the Archean time period. So these are the things which were recorded from 3.5 to 3.3. GA means the gigaana, or you can convert into the billion years, or you can call 3,500 to 3,300 million years old microbial mat from the Barberton Greenstone Belt of South Africa. So these are the different structures, and they are also known as laminites, and are also known as microbially induced sedimentary structures. These are from the Western Australian region, which is again the 3.48 billion year old. Similar structures are we are trying to find out in the India Archean successions also. This here I am showing you the comparison. These are the recent one on the seashore, and the similar structures are found in the Pongola supergroup in South Africa, which is again from the Archean succession, that is 2,900 million year old, or 2.9 GA or gigaana. So you see how similar these structures are from the recent activities, and these are all in the rocks, what you are seeing here. Now coming to the one major thing, how the oxygen came into being. As I mentioned that the oxygen was not there since the beginning. So there are people have studied several and studied the mineral composition of the rocks, and then came to the conclusion that there were different puffs there. Uh, but if you divide into several parts, then there are the 5 stages. The stage 1 is starting from 3.85 to 2.45 GA. That was the time period when the Earth was entirely anoxic. Then is from stage 2.45 to 1.85 GA. The level was very mild. Shallow sea was mildly oxygenated, but the deep sea was anoxic. Again, the stage 3, which is start from 1.85 to 0.85 billion years, sea was mildly oxygenated. And in stage 4, from 8.5 to 0.54, a rise in atmospheric level, and that was about 0.2 atmospheric level of the present day. And after that, that is known as the Phanerozoic time period, when the oxygen was there in the environment, and that gave rise to the complex form of life. So that is in the stage 5, which start from 0.54 to present day, and the oxygen level rose from 0.2 to 2.3, and then it attained the present value, except in the certain part where the Carboniferous. Now coming to the Archeans of India, and the cratons are those regions which are not disturbed, or the very primitive parts. And India is in a mosaic of different cratons. You see, one is the Dharwar Craton in the South India, the Bastar Craton in the Central India, Singhbhumi Craton in the western part, and Bundelkhand Craton in the northern part, and the Aravalli Craton in the northwestern part of the India. So this is now joined together, and that constitute the peninsular part of the India. So these are the craton, and then there is a granulated terrain in the southern India. So these cratons are the hub of activities, because if we have to study the Archean part, all of these cratons are from the Archean age. And if we have to study the sedimentary processes or the biological form, we have to explore these regions. And let me mention here that most of these cratons are constituted of metamorphic or the igneous rocks. There are very little bit of sedimentary successions, and those sedimentary successions are targeted for study of the biosphere. Once again, the distribution of the various craton, and since most of the record which I am going to present today is coming from the western part of the Dharwar Craton. This is the part which I have shown you in the earlier slide, which is again divided into the western Dharwar Craton and the eastern Dharwar Craton. Now coming to the craton, as I mentioned, these are the oldest forms. So if we have to estimate or understand that how old they are, so these are the dates uh, um, conceived by different workers, those who have dated the by the geochronological ages. The eastern Dharwar is 3.4 to 2.7 GA old, western Dharwar is 3.2 to 2.9 GA old, southern granulites are 3.5 to 2.4 GA old, and the Bastar is 3.6 to 2.5 GA old, Singhbhumi is 3.6 to 2.5 GA old, Bundelkhand is again 3.5 to 2.8, and Aravalis are 3.3 to 2.5 GA. These cratons are very well developed during the Archean period. We do not get the hidden rocks anywhere in the world, but we have sometimes zircon grains. And incidentally, India has also found some of the zircons which are around 4 billion year old, approximately of that age. So those zircons are found in the different rocks, but they are not the original rocks. They are the secondary rock which were melted and once again poured on the earth and then got deposited. So there are the zoning, and if we the geochronologists who date it, then they see that the internal part is the oldest part, and that is how they come to the conclusion that the hidden during the hidden time also there were some rocks which were having the zircon grains. They were weathered due to the erosion and then got redeposited, and so that is why we have a distinct rocks of these ages in the Indian Craton, but we do not have a rock of Hadean ages. And this is then the comparison what is we are finding here. If you see that in the western part, what is happening is that we have a very good sedimentary succession here. Here in the eastern part, most of the rocks are of igneous origin or the metamorphic origin. Here, whereas in the peninsular gneisses, unconformity is there, quartz pebble conglomerates are there, and then some subordinate sediments are also there. And we are looking these subordinate sediments in which we found the microfossils or the stromatolites that I will be showing you. If you remember that what type of stromatolite I have shown you from the Australia or the Pangola group in South Africa, or in from the Greenland, they were very small, very rudimentary. Whereas in comparison to that, the Indian stromatolites are very well preserved in the Archean. Although the Indian Archean succession, sedimentary successions are younger, they are on the Neoproterozoic, that is younger than 2,800 million year old. So these are the some of the cyst belt we commonly call in the different successions of rock as a Chitradurga cyst belt, Shimoga cyst belt, and the Sandur cyst belt, and they have the ages. And we have recorded almost stromatolite from all the three cyst belts and microfossils from Chitradurga and the Sandur cyst belt. Once again, I would like to show these are the pillow lavas which are exposed in the Chitradurga district, and these are supposed to be the volcanic rocks which were just pouring out in the very close proximity to the sea, and then they got cooled down. And here these are the volcanic rocks which have some covering also there. These are inhabited by the earliest organisms, and if we prepare a thin section, then sometime if we are lucky, we get very good microfossils from these surfaces. And this particular activity is taking place even today, although these are the organic igneous rocks. But what is happening is that when they cool down, then the first thing which attack these things or colonize these rocks are the microbes, and those microbes are the first or earliest inhabitants of these particular rocks. This is the one show, Mr. Close-up of that particular rock. You can see how these particular rims are there, and the rest of the things is igneous in nature. Whereas the rims are very close to be the formed by subsequent weathering and leaching and different phenomena of the sedimentary succession, and these are attacked by the microbes. Now here, for your comparison, I am showing you the Archean stromatolite from the Varavana group and the Archean stromatolite from the Dharwar supergroup. This is an Archean one. Here you can see from the Dharwar supergroup stromatolites. This is the field succession where we note that there are very good exposures of stromatolite rock and the dolomite. This is a dolomite rock in which we find the stromatolites. This is again another type of stromatolite that is known as an egg carton shaped stromatolite, which we have recently recorded from the Dharwar Craton. And this type of stromatolite has also been recorded from Estuary Pool in Pilbara group of Australia, or that is an Australian succession. This is a columnar type of stromatolite. If you see those earliest stromatolite for the laminated form, but subsequently they developed a domal shape, then they developed a column, and their column was also naked. If there is no wall, then they are known as a naked column. And this is an evolutionary tendency that due to the evolution, they used to themselves very safe, then they used to develop a wall. These are again the structures, Archean stromatolite from the Dharwar Craton. You see different types of stromatolites from Shimoga cyst belt. These are the three cyst belt, as I mentioned, the Chitradurga, Shimoga, and Sandur cyst belt, and all of them are having stromatolites. You see very good and well naked columns are present. In the Archeans, we do not get the wall structure in the stromatolite because there was no competition by the different other microbes which can feed on these microbial laminated structures. That is why they were almost naked in the shape. But if we go in the Proterozoic or in the terminal part of Proterozoic, we see that they have a very good distinct wall structures are also there. You see the different types of stromatolite from different cyst belts. Again, that's from the Chitradurga cyst belt stromatolites. They are very well preserved, and in comparison to the other parts of the world, Indian stromatolites are very well preserved. So I'm showing you, this is the work of last year's when we made a field visit to this particular locality. We have documented these type of stromatolites from this area. You see, now you can see, and if you compare with those one from Australia and the South Africa, they are excellently and well preserved. Now coming to the, what is the level of oxygen and how we measure it. As I mentioned that in the early part of the Archean, there was a very little oxygen. If you see, this is a curve which has been made based on different minerals which are occurring, because some of the minerals are very easily oxidized. And if we get those minerals in our successions, that shows that, that means at that time there were no oxygen available in the atmosphere. Otherwise, that would not have traveled from the hinterlands and got deposited otherwise and got oxidized in the course of movement from hinterlands. So if we get the pyrite, if we get the uranium, these are some of the minerals, and the manganese, that suggests that yes, in the earliest part we were not having oxygen in the atmosphere. Whereas if we are getting a lot of banded iron formation, that suggests that yes, oxygen was available in the atmosphere, and that is how the iron was getting precipitated in the course of action or during the weathering, and subsequently mixing with the water in the sea, and got deposited in the layered form that is known as banded iron formations. I will show you here. You see, this is the pyrite in the limestone of the Bani Vilas formation of the Dharwar supergroup. So we are getting a very good pyrite, which is unoxidized here. So why we are getting? Because there was no oxygen available in the atmosphere, and that is how they were released from the hinterland and got deposited where the limestone was getting deposited. The pyrites are formed in an anoxic environment, as well as they got oxidized in the oxygenated environment. So this is an terrigenous pyrite which is occurring here in the limestones, and very well preserved. Very good crystals are there. So that suggests that oxygen was not present in the earliest form. And this is one graph which shows that how the oxygen came into being, and then when the plenty of oxygen was there, whatever iron was dissolved in the seawater got precipitated in the form of oxides, that is known as the banded iron formation, and most of them got deposited before 2400 million year old. We know the great, that is known as the great oxidation event. So means if you see, this is the entire part which is whenever there was an oxygen available, iron was getting deposited. But there is a one more peak here which says that there was around 1.8 billion year time period. The oxygen was not there in the atmosphere, or sucked by some phenomena, and then that oxygen, no, not sucked, but it was consumed by these banded iron formation suddenly, and there was a whiff of iron there, and lots of banded iron formation got deposited around 1.8 billion a year. And after that means almost all the iron was deposited, and in subsequent earth period of the earth history we do not have banded iron formation. So the last banded iron formation came, this is a major banded iron formation that was around 1.8 billion a year. Then when we see this different types of sedimentary structure, when we prepare a thin section, as I mentioned you earlier, we used to get different structures which may look like the organic. This is the big question. It happens whenever any paleobiologist demonstrate anything, then it is questioned whether they are the fossil or not the fossil, or are inorganic structures. So these are from the ISWA Greenland belts that was reported in 1978 by Fluke, and this was variedly debated throughout the world, and people are skeptical about these things whether they are the biological or not. But these are putative microfossils of the oldest from the oldest rock of the world, that is a 3.8 billion year old rocks. Then comes from the South Africa. The people have studied, the Bill Schaff has studied, and these are the fine narrow filaments they have recorded from Krumberg formation. And similarly from the Western Australian apex shirt, they have documented these type of features. So you see these, how these features are very faint, and it's very, you need to have a very trained eyes to document and distinguish these features as a microfossil, because you see this particular filamentous form has been documented in 4 or 5 parts. This is the one part, then in one second focus, third focus, fifth focus, and sixth focus, and then they have merged it to reconstruct this entire filament. So similarly here, this one which you are seeing is on one part, but if you see the at a higher resolution, you will find that this particular specimen is made up of 35 slices, which were taken at different focal length. So finding fossil is very difficult. Identifying fossil is difficult, and establishing the fossil is Archean fossil is very difficult. Here we are once again we are lucky that in Indian case, Indian Archean microfossils are very well preserved, and that as you will see, these are again different microfossils recorded from different other formations of South Africa. Here the broad tubular filaments, now it is a very well preserved, but now come here, and if you see, this is the one stromatolite part, and if you have a chert here, then you prepare a thin section, and if you gradually keep on changing the magnification, then you will find that they are made up of different types of fossils. These are the Warabuna microfossils taken from the chert. And here our Dharwar microfossils, you see how well preserved they are. When we have taken the chert samples from the very close to the Sandhu cyst belt stromatolites, we have noted different types of fossils. You see the preservation level of fossils is very good. They are all preserved here in the chert, and these are the chert from where we have taken the samples and prepared the slides. You see the level of preservation is good, and we call them as the Formidella tenui and Formidella sandorense. You need not go to the naming, that is an altogether different business. But here you can see the different types of fossils which we have recorded from the Archean microfossils. Once again, you can see this cluster of microfossils are present, and if you gradually keep on changing the magnification of your slide, then you will see that these fossils are from this particular locality. Once again here, what is happening is now that when we go to the Precambrian and even in the Phanerozoic, Proterozoic part, we have very good fossils in comparison to the Archean ones, and those fossils are very well comparable to the living ones. Here I am showing you a modern oscillatoria, and this is the fossil oscillatoria from the Bitter Spring Formation of Australia. Then here the again modern oscillatoria and the fossil oscillatoria you can see here. So in the fossils, this is the one thing. The cyanobacteria are the most conservative. It is estimated that they have developed sometime in the Archeans, and then they are still maintaining their morphology, shape, and function as they were at that time. So these are known as a conservative evolution. This is known as a conservative evolution, and the fossils are same as they were at that time. And in the Darwin's terms, we will say who is the most successful. He mentioned that survival of the fittest, and I will say the cyanobacteria were the best thing, and they were the fittest form because they originated sometime in Archeans, and then they still are continuing. If you go to the any pond or any part of the sea, you will find that different types of cyanobacteria are still flourishing in those water and sediments. Now I'm turning to this knowledge to the further astrobiology, what has happened then and how this has developed. This is the new discipline which suddenly came into being almost 24 years back, when there was a report of uh, organism from or life form in the Mars meteorite that was discovered in ALH 8401. At the time, there was a conference in 1997 in the Senckenberg Museum where they were discussing the paleontology in 21st century, and this term was coined at that time, that we must study the astrobiology. On the entire Earth history, 5-6 part encompassing the life history was ruled by microbial inhabitants. As I mentioned, that 5-6 part is Archean. The rest, 541 million year, is the only Phanerozoic which has seen the entire evolution. But for conservative estimates, the microbial life inhabited the 5-6 part, and many of the available earliest evidence of life are disputed and suggest that we need to know more about biological origin of the microbial fossils. As I mentioned, that it is always questioned when you report Archean microfossils, but we must not get disheartened, and we must keep on working, and then we will see that lots of scope is there. Now, what we expect from the astrobiology? It is a scientific study. Of course, it is a scientific study. It seeks the distribution and density of life in the universe. It is a fledgling science. How do habitable worlds form and how do they evolve? How do the living system emerge? How can we recognize other biospheres? How have the Earth and its biosphere influenced each other over the time? And how rapid changes affect ecosystem? Means these are some of the questions which come to our mind regarding the Archeans, and they hold true for astrobiology as well. This is one of the another question about the exploring the role of serpentinization in the evolution of early life and their metabolic processes in early life. For that, we have once again a very good site in India, that is known as the Sukunda belt in India, where we get lots of serpentine. And there is a one more area in the Andhra Pradesh where we find the serpentine in plenty. So we can study all those things. That is a natural laboratory. Then then comes the question, the where we will find the clue for astrobiology or the fossil from the for the astrobiology. Of course, we cannot find the fossils in igneous rocks. If we cannot find the fossils which have undergone the extreme heat, means 600 degree centigrade or 300 degree centigrade and above. But if it is cooled down igneous rock, as I mentioned, in the pillow lavas, on the periphery of the pillow lavas, we can expect to get the fossils, early colonizers there also. And similarly, in the Archean rocks, we need to separate the organic matter soup from the Archean rocks. Their chemical characterization and laboratory bed reactivate to understand the primary organic reaction and their fate. This will help to understand the possible mechanism that may have produced early sensitive, super sensitive organic structures. Then how it is linked with Archean life and astrobiology. Modern version of panspermia is steadily grown into concept of astrobiology. It is far beyond the paleontology, but it provides a link between the two. That if we can look for the fossils from in the Archeans, and then we can recapitulate all these things in the different atmospheres like the Mars or other planetary bodies. Then what is the goal for the astrobiology? To locate and interpret evidence of former life, draw support from diverse group of scientists. It is indeed a multidisciplinary approach, such as paleobiologists, geochemists, and mineralogists. They all work for understanding the early life forms and how the early organisms were drawing the energy from different chemical reactions. Then these are the issues, as I mentioned, the nature of life on Earth, discrepancy between the maps of molecular biologists and fossil record. What is happening is that the molecular biologists suggest that the earliest eukaryote came into being around 2.7 billion year old, but actual fossils we used to get a billion year later, around 1.6 billion in old rocks. So this is the discrepancy which we note. How many types of tree of life exist and niches where life can survive. At one point of time, people were thinking that the only sedimentary rock which have not undergone a high-grade metamorphism are suitable for the study of microfossils or microbial life. But now today we have seen that very close to the mid-oceanic ridges, we have a very thriving life is found. So we can see similar conditions in the Archean successions or in the other planet, on the other planet also, where we can look for the evidence of life. And then determination of the lower limit of size is what could be the size, means 2 micron, 5 micron, or submicron level, and what is the upper temperature limit of the life, means whether we can expect to get the life at more than 70 degree, 80 degree or not. These are some of the issues for the astrobiology also, because we do not know what is the temperature on the other planets where life could have survived the high temperature. The other issues are the three super kingdom of life on Earth has very ancient origins and should be part of investigation for evidence of life on Earth and elsewhere. And similarly, the biodiversity is also we are looking for. Means suppose we are looking for the oxygenated environment, so we will be looking for the microbes which are generating or exuding the oxygen in the atmosphere, or some which are consuming the oxygen. So these are the animal world as well as the plant world, and some microbes which are using the sulfur as an energy source. Some are using a methane as an energy source for survival. So these are some of the microbial world, and these are the this is the type of biodiversity which we are looking for in the astrobiology also. Then what we do, we study the extreme environments. we study the deeply buried subsurface sediments in the fracture of deep-seated igneous rocks, in the frigid Arctic and Antarctic lakes, in the boiling thermal springs, in acidic and alkaline lakes, and around the mid-oceanic ridges. As I mentioned, that now the people are also studying the mid-oceanic ridges for the symptoms of life or the thriving life which are now documented. Then the paleosols, evaporites, hydrothermal minerals. These are some of the things which we are looking. And even the desert varnish. This is the new thing where we are looking for this particular type of feature which develops on the sands of the desert, and then the varnish, that particular varnish is supposed to be made by the activity of microbial remains. Then what is our approach? All possible techniques are employed for the search of evidence of early life. Characterization we are doing morphologically, and as well as the chemical studies or the isotopic and organic, and then the composition and history of planetary atmosphere. We are documenting biological information. We are retrieving form in different forms as a signature and trends of isotope and the hydrocarbon molecules which we are looking for. So these are some of the techniques which we are famous people are
I think Dr. Sharma's line has dropped. So please bear with us for 2 minutes. I will just try to recover his line. Yeah, we are just trying to get back Dr. Mukund Sharma. Meanwhile, I would encourage the participants to think of some good questions, and you can list them in the Q&A section here. And just give us 2 more minutes. I think he's facing some internet outage in his office. So until then, maybe in 2 minutes of time, hopefully we should be able to resume. I encourage everyone to just post some questions, and we will be back with you very soon. Thank you.
हो गया लाइव चल रहा था यूट्यूब पर, ये कहाँ से होगा भाई? यहाँ से स्क्रीन आपको शेयर करनी है ना स्क्रीन? हाँ, शेयर स्क्रीन। हेलो हेलो, हाँ हाँ, are you able to see it?
Yes, sir, we can hear you now.
Okay. Just few slides. Sorry, there was some internet problem. Okay. So, as I was mentioning that it has a better appeal. Was this the last slide you were seeing? Or
So
shall I go back?
One slide previous.
This one? Or okay, I start. What is the scope of astrobiology? The concept is new, as I mentioned. This is not more than 24 years old, but it has a very big and outreach appeal, because what has happened in recent times, I think 10 days ago, there was a news item that the NASA people have found phosphine in the environment of Venus. So this was the news, and people are thrilled to know that there are some signatures of life forms or life molecules are there. Similar things were reported by exclusively the Indian group, reported in Astrobiology 2 years back in 2018. They have also found out in the clouds of Venus that they have noted the presence of phosphine. So this has a very good appeal, and the leading societies are having their astrobiological centers. And we are fortunate that in India, the ISRO as well as some private universities are also taking interest in establishing the center, and MIT University has taken a lead and had already established a center of excellence in astrobiological. So this is going to be a new venture and a new scope for studies for different people, those who are willing to do or pursue the science. And son, what are the now issues in exploring early life? We want to study the earliest part, that is a 3,500 million year old history of biosphere. We want to record earliest possible evidence of life, that deciphering the signatures on what type of those life forms were, and record the early experimentation and consequent evolution in biosphere. Some of the forms, what is happening is that sometimes we get the forms which do not look like very similar to what we know today. They may or they may not be the biological entities. They may be altogether a different form, but they may as well be a biological form. So we have to establish and find out that the was that particular form was an early experimentation of nature, and consequently they have any role in the evolution in biosphere. And then we have to find out on the trace the antiquity of archaebacteria, methanogens, cyanobacteria, eukaryotes, metaphytes, and metazoa, because all these things have led to development and evolution of complex life forms in the Phanerozoic time period. Then these are the opportunities. As I mentioned, that we have a very good cratonic parts, cratonic shields in India, where very few remnants of sedimentary rocks are there. But those sedimentary rocks are very precious for all of us, for the entire humankind, because those who want to study, they have to come to here in India. Likewise, if I mentioned that if the Japan, every country is not having the cratons. Suppose Japan has everything. They have the people, they have the machine, they have the willpower also, but they do not have the Precambrian rocks. If they want to study the earliest early life, they have to go to the South Africa or to Australia or to India. So we are fortunate. What that means, we are fortunate. We have the craton parts, and then we have Paleoproterozoic and Mesoproterozoic rocks are also there, in which we can see how the forms which evolved during the Archean, they have transformed and became complex form on the eukaryotic forms. So these are some of the things which are areas which people are studying, and the current endeavors are we are studying the Archean Stromatolite. As you mentioned, that last year we have also gone to the Dharwar Craton, and we have documented very good number of stromatolite from there, that area. We have found out new type of stromatolite which we have documented, that is known as the egg carton shaped stromatolites. And then we are finding life forms in the extreme environment also. Like extreme environments, what is that extreme environment? Today we know that life cannot sustain in the hot temperature beyond the 70 degrees centigrade, but we have to find out the signatures in the hottest springs. We have to find out what is the best thermodynamic calculation for the existence of rock. And that is, once again, we are fortunate enough that we have a large number of hot springs in India, where we can look for these falls type of life forms. And then these are the our objectives, and these should be our objective, which we are if we want to pursue the astrobiology in India. But I will show you now. These are the things where we have to find out the life in extreme condition, that are the hot springs, permafrost, glaciers, saline lakes, paleo lakes, cold or high desert soils. And fortunately, India is having all these environments where we can study this type of life forms, which can be very helpful in understanding the astrobiological signatures. I will show you where we can go. This is the Pangong So Lake in Ladakh area. Here, there is a Puga hot water spring where we have studied, and we made some contribution for understanding the life forms. These, you see, these are some of the stromatolitic forms which we have noted there, and the work is in progress here. They have the slimes here. So the temperature is high. We cannot expect to get the cyanobacteria here, but some other types of microbes are possibly occurring in this particular field. And we can find out that these may be methanogens or some hot water loving microbes. You see, the similar type of thing has been documented in Altatio in Chile, where people have documented very fine microbes in them. This is an structure. This is the living form here. This is the matte structure here, and these are under the scanning electron microscope, where they have shown you the filamentous streamers are present there. Then comes the people have documented, means the NASA is taking a lead here. They have documented and mapped certain areas, particularly the home plateau there, home plate, where the opaline silica they have recorded here, and the similar things have been noted in Altatio again in the child locality. this type of forms, this is a from the Mars locality, and this is from on the Earth. And on Earth we have similar type of fossils in the Kalandigi group. You see, these are the Asperia digitata polished slab, where you can see the microstromatolites are there. If you see the previous slide, these are the microbial stromatolites of the present day. This is what we are seeing in case of on the Mars. These are some of the regions. Then we can see similar things in the Sambhar Lake locality in the Rajasthan, where different types of salts are there. And if we study the microbes occurring in those playas, then we can find halobacter bacteria, salt-loving bacteria. And similar thing we can see in the Sokar area also. You see, the lots of salt is there, and microbes can be studied here. You see. Then this is the Panamic locality where the incrustations are there. And if you prepare a thin section here, then we can document different types of microbes present in that particular locality. Once again, I am showing you the people what they are looking for on the Martian surface, and this is on the Altatio once again. The different types of salts are present there. So similar things are we are looking in the Ladakh area also. We can find out all these things which are very relevant for astrobiology. So you see the salt panes are there in the Rajasthan locality, and then once again there is a one suitable locality in the Lonar crater, where the crater which was formed by the meteoritic impact, and now it is having a very rimmed structure, and on the rims we can look for endolithic algae, or the earliest microbe which might have inhabited these particular volcanic rocks here, because this is the only crater which we can say is occurring on the volcanic province, or on the surface peninsular regions volcanic province. So here we can test that particular hypothesis, where we have seen different types of microbes in the pillow lavas, on the rims of the pillow lavas. This is a one attempt made by our scientist and the artist here, where we have shown you lots of bombardments, and then how the life arose here, and then the stromatolites, fumaroles, microstromatolites again, and then the animal forms. So this is the one lesson where we can see that Archeans and the knowledge of Archean life, Archean forms can help us in understanding and designing our experiment for the astrobiology. This is all what I have to say. And once again, I'm thankful to the Center for Excellence in Astrobiology, MIT University, which has given an opportunity me to discuss all of you. And this is a good thing that they have taken a lead. I admire that part. And I am supported by my fellow colleagues in my laboratory, including Viru Kant Singh, Santosh Pandey, Arif Ansari, Yugma Shukla, Shamim Ahmad, Yogesh Kumar, and Divya Singh. And over and above, the technical assistance given by Archana Sonkar is highly appreciated. This is all what I have to say in this presentation. Thank you.
Thank you very much, Dr. Mukund Sharma. I think we had an exciting journey.
Yeah, I'm sorry that there was some break in between.
No problem,
but
sir.
Anyway,
in these difficult times, I think such things are inevitable. That is not a problem at all. And I must also add that even though our center has taken the lead, we will not be able to achieve all our plans without strong hand holding and support from BSIP, and we really acknowledge the vast experience that BSIP holds both in Precambrian and Quaternary, and through the connections with yourself and Dr. Benita Madam, and possibly other scientists who we have worked with. I think it will be a joint collaborative effort to establish astrobiology and for the students here in India. So maybe we will have time for, say, about 10 minutes. Let's just spend some time, because I'm seeing some nice questions have come in, sir. So
yeah,
if you don't
this
mind,
is the one question here.
Yes, sir.
I will be happy to answer.
Feel free to choose the question, sir, whichever one you would like. I think you can see them as well.
The first question is, are these rocks formed only by cyanobacteria? Are there any other bacteria also, any kind of other slime forming bacteria or biofilm? First, let me make it clear that these rocks are not formed by cyanobacteria. This is an interplay of sedimentary process as well as the microbial process. And the earliest stromatolites are not formed by the cyanobacteria. It is in the later part of Archean that those rocks, in those rocks we find the cyanobacterial fossils. Whereas the earliest part, they were the laminites and formed by different types of sulfur bacteria, or the methanogens, or the other different types of bacteria were involved in the formation of laminated structures. That is why in the earliest part of Archean, we get only the laminated or small domal forms. But as soon as these forms evolved and gave rise to the cyanobacteria helped in the precipitation of carbonate rocks. And then the large columnar stromatolites or the branching forms came into existence. So, Dr. Renita, it is clear that all these rocks are not formed by cyanobacteria. Rocks are not formed by the cyanobacteria, but stromatolites are indeed and formed by the interplay of cyanobacteria as well as the carbonate precipitation. But other types of bacteria and the slimes have also participated in the formation of stromatolites. But sedimentary rocks are formed by all different methods. Then I don't see any question here, but I would like to take questions
Yes,
from
sir.
anybody.
I think, sir, maybe because you had got disconnected, so the list has not come to you. I will read out the question, sir. So
Sure,
Gaurav
sure.
Yadav has asked, can the microfossils of extremophiles be revived in the lab?
At present, our knowledge is not sufficient to revive the things as far as the fossil is concerned.
Right, sir.
So far we have we do not have any technique known to us where we can revive the fossils in the present form. And I think this question is emanating from his knowledge of the Jurassic Park.
Yes, sir. Possibly, possibly. Okay.
Yeah.
Another follow-up. Another question is, where can the stromatolites be found in India? I know you mentioned some regions, but maybe if we can connect them to actual states or regions of our country.
Yeah, yeah, sure. I can say that almost every state is having Precambrian rocks. Every state, and in those sedimentary rocks we have a definite stromatolite for. May it be your Maharashtra, Andhra Pradesh, Chhattisgarh, Madhya Pradesh, or the Himachal Pradesh, or the Rajasthan, or in the Karnataka. But we do not have a stromatolite record from Kerala, and we do not have a stromatolite record from Tamil Nadu. But we have a stromatolite record from Jharkhand. We have a stromatolite record from Bihar, Uttar Pradesh, means almost Uttarakhand, means almost everywhere we have a record. Even in the Sikkim, we have lots of record from Arunachal Pradesh. So what you have to look for the Precambrian rocks, and then look for the carbonate rocks. And in those carbonate rocks, there are excellent stromatolites are present there.
Sir, have you found stromatolites in Sri Lanka as well?
So far not recorded from the peninsular region, but in the sea region between Sri Lanka and Talaimannar of the Indian side, there are some living stromatolites were recorded by Deshikachari. Microbial mats, I can say, not the stromatolite, but the flourishing microbial mats, which are akin to stromatolites, were documented by Professor T.V. Desikachari of the Madras University,
Right, sir.
but not from Sri Lanka.
Thank you, sir. Next question is from Neha Potnis. She's asking, how do we predict and confirm the age of the rocks fossils that we discover? And second question she has asked is, what is a laminated structure?
Okay. The dating is altogether a new science. Not new science, but you can say it's a different science, where the geochronologists use the tool, that is the igneous rocks, particularly the granites, or the rocks which are having a very good amount of zircons. And zircons are dated with the help of different methods. and on that the decay constant, on the basis of the decay constants, they determine the age of that particular rock in which those zircons are present. So this is the technique, and we seldom we date the actual sedimentary rock. What is happening is that we get the igneous rock underlying the sedimentary rock, or it is overlain by the some igneous rock, and we date those rocks. And on the basis of those datings, we presume that suppose the overlying rock is 10 million year and the underlying rock is it's a 13 million year. So we can say that the sedimentary rock occurring in between the 10 and 13 million year old is older than 10 million old or younger than 13 million year old. This is the one concept of the dating. But now the people have also started dating the sedimentary rock directly. That is the black shale, and in some cases the glauconite is the mineral which is dated. So with the help of that, people have tried to explain the exact date of sedimentary rock, or what has happens that if there is some volcanic activity in some part of the world and ash is coming to that particular region where this particular sedimentary rock is being get deposited, so we date that sedimentary activity, and then we correlate the sedimentation as well as the volcanic activity, and then tell the age of the rock. And
Right,
then
sir.
what is the laminated structures? Laminated structures are those structure wherein there is an interplay of microbes. That may be a cyanobacteria, that may be some other bacterial forces, microbes that gave rise to the formation of laminated structures. And if you see under the thin section, even in the living form or in the fossil form, we can get the microbes present there. So these are the laminated structures are indirect evidence of biological activity in any sedimentary rock.
Right, sir. Thank you, sir. So next question I will just try to modify a little bit is that basically to ask, suppose we are now observing and making visual and spectral records of the different kinds of rocks samples and mineral samples on Mars. So how confident are we, or how detailed is our understanding on Earth that by looking at, just by studying and noting the kind of rocks and minerals on Mars, that we can try to say that these are the possible candidates of microorganisms that could be microfossils that could be present inside it. So that correlation between the rocks and the kinds of microfossils that have been found on Earth, how confident are we that we can take that knowledge and translate it to what we are finding on Mars?
The level of confidence is yet not 100%, but we can say that when we see that similar forms, morphological features on the Mars and in the rocks, as well as when we see them on the Earth in Archean rocks, as well as in the living form. Because I have shown you one example where they have seen the opaline silica on the Altatio in the Chile, that is a living form. And when we see the similar forms in the Kaladigi in the Karnataka, and when we see the similar things on the home plate on the Mars. So that is how we correlate or we join the dots. And once again, I will say that the confidence level is as good as we can believe, because
Right.
there, until unless we get that particular sample from Mars and study in the laboratory, we cannot come to the 100% conclusion that yes, those opaline silica which are occurring on the Mars having the similar morphological feature as those of the Altatio in Chile or in the Kaladiki or in different other forms, they have the same type of origin and they were formed by similar organisms. So you can say at present we can say the 50% is confidence level, but we have to join these dots. Is the Mars similar analog, living modern analog from on the Earth, and similar features in the Earth history in the Archean rocks. And if we are able to join these dots, then yes, we can be fairly good confident about those features present on the Mars, made by organic remains or the microbial remains.
Right, sir. Sir, this is another question. Other than the cyanobacteria or halobacteria, what are the other kinds of bacteria that are more dominantly found in microfossils, and why is it that cyanobacteria has achieved this conservative evolution? What is the reason behind that?
No, as we can find all types of microbial remains, means that should be of the same pH conditions. Means suppose some are alkaline, some are acidic. So those who are occurring in which are occurring in acidic environment, so we can understand to find only the acidic environment microbes in that particular laminated structure. Suppose we are finding alkaline environment, and in that particular alkaline environment, whatever laminated structure is present, we can hope to get the alkaline microbes in that present. May it be cyanobacteria, may it be halobacteria, may it be anything else, thermophiles also, and we can say that some other microbes also may be present. This is
Right,
not
sir.
only the lower forms, cyanobacteria or the still bacterial forms. We have some complicated forms like in some cases we get lots of pustules also, siliceous pustules in the stromatolite. We get all these things. As far as that last part of your question is that why this the cyanobacteria is continuing, their conservatism is their success, and what is happening is that they can survive in different environments. So they survive all type of environment. They normally they are found in a very neutral environment, but they can sustain up to a very feeble alkaline as well as acidic environment also. That is why they are continuing there. But what happens that when some organisms, the animals are present there in the vicinity or in the surroundings, which feed on this cyanobacteria, in that particular locality we are not going to find out. But wherever the environment is favorable for them, they are still flourishing, and that is the success, that is why they are continuing from Archean to present day. Their
Right, sir.
conservatism is there uh, in surviving in all types of environment. Means they can survive in high temperature region also, very cold region also, very alkaline region also, and in acidic region also. This is the one region. Otherwise all other organisms have very great preference for certain type of environment, and that is why they are confined to that particular region, whereas cyanobacteria can survive in any region. That is why we can find them in permafrost, we can find in the Arctic, in the Antarctic regions, or in the glaciers, or in the saline pools also, or in the alkaline.
Right, sir. So two more last questions we will take. One is a science question and another is a education
Sure, sure.
related question. Science question is from Gaurav. He is asking, what is the method to identify species presence in microfossils? Is it the 16S RNA sequence similarity method? And I'll also ask you the education
Yeah,
question
here
that in the initial undergraduate years, what should be the focus for student if they want to study geobiology of extreme environments?
let me make it very clear here. As far as the present day definition of species goes, that suggests that until unless we see that it is interbreeding, we cannot define the species correctness. So in fossil, we cannot ascertain whether they were interbreeding or not. So whatever the species name which we are giving is just an artificial name. Suppose I will give an example. Today we know that the Antophysalis grows in a freshwater environment, and and it has certain micro morphological features. And if we are getting the similar morphological feature in the Precambrian rocks, in the chart of different Precambrian rocks, we will say that these are the analogous to Antophysalis, and we will give them name as the Eoantophysalis. Eo means the older, Eoantophysalis, but we cannot definitely ascertain that these are true representative of Antophysalis. Likewise, as I have shown you the oscillatoria, for fossil oscillatoria, we call them as the paleo oscillatoropsis or oscillatoreopsis or paleolingbia. If we are getting a fossil of lingbia, which is having a morphological feature too similar to the present day lingbia, we call them as a paleolingbia. So these are the traditions in naming of the fossils, because we cannot certainly definitely say that yes, they are the true representative of the older, or means the younger forms. But this hold good only for the Precambrian. If we go in the younger forms, means the tertiary forms or the Holocene, where we can truly establish the species level occurrences in the fossil record. But it cannot be applied in case of Precambrian microfossils. So the question does not arise how we are going to ascertain. It is entirely based on the morphological features.
Right. The second question is
Uh,
regarding the student
yes,
asking about geobiology.
yes, yeah, geobiology. If they are very interested, then I think yes, certainly they should take the biology and the early geology courses. In the geology courses, people study the sedimentary rocks. People study the fossil part, fossils also. So this is an amalgamation of biology and the geology, which you can in the Indian system you can study at the graduation level. So you will have to opt the geology as such. And I think nowadays in botany on the geology, very little part of fossil or the paleontology is taught. So it is only realm is the geology. You have to study the geology, where you can have the option of studying the paleontology at the senior level or then the post graduation level. But certainly, indeed, you will be taught paleontology in your undergraduate classes. So the paleontology and the geology is the subject which you have to opt if you want to study the geobiology.
So one more connecting question has come from a student. She's final year MSc geology student. That what is the scope of geobiology in India?
Geobiology as such is now, since you people have also started, and some other institutions like Pune University has started, and the ISER Pune is also having um, a study center in the chemical division as well as in the geological division. So I think it is a pretty good for doing the research work after doing your post graduation. And we are hopeful that ISRO will also come forward and grant projects or establish some center for study of astrobiology or the geobiology, uh, which I think are more or less the same thing,
Right, sir.
because
Right,
there are
sir.
the three journals which are coming for uh, the research work publication in the International Journal of Astrobiology, and the Geobiology. These are the
Yes,
three
sir.
journals that shows that there are lots of people are doing research, and Indians can also communicate. So yes, we are behind schedule, but certainly we will find time, and our government agencies and the institution will also come forward to initiate this particular course in their universities. But it is not like that, like the computer sciences where everybody is doing computer and getting job. But yes, scope is there.
Right, sir. Right, sir. I think that brings us to the end of the Q&A. We have answered all the questions, and the session has been recorded and watched live by a nice class of about 36 students. And we'll also be having recordings that we'll be sharing with the other astrobiology course students. So with
Okay.
this, sir, we would like to thank you for your time today morning on a weekday, taking out time for this. And we hope to have more such sessions in the future as well, where you can share some more perhaps some experiences from the field when you have been doing a lot of field work, and what are some things that, because we need to also attract students to come out of their labs and classrooms and enter into the field environment, and there are very few opportunities for students in India to do that. So like maybe next time we will talk a bit about that as well, sir.
Yeah, certainly I would like to take them to the field also and show the things.
Yes, sir, definitely, sir. Thank you very much, sir. And thank you to all the participants for joining today. And next monthly webinar will take place in November. We've had an early start this month. And most likely, Dr. Binita Fartial, also from BSIP Lucknow, she will be giving the lecture. The date and time and topic will be announced soon on our website. So please subscribe and stay updated. And hope everybody has a nice day ahead and evening ahead. Thank you, everyone. And I'd request Dr. Mukund Sharma if you could just stay online. I will just stop the recording as well. Thank you, everybody.
Okay.
Okay, sir. Recording has stopped, sir. Thank you so much, sir. I think this went well.
Okay. Yeah, but there was some interruption in between. There was I don't know what has happened.
No
on our
problem, sir. No
side.
problem. That happens. That is not a problem, sir.
And
And
that was for some five minutes,
ha,
4 or 5 minutes.
5 minutes.
There was
5 minutes.
no.
That's okay,
Right.
sir. I was just engaging with them, asking some questions to the audience. So we manage that. Not a problem. Right, sir. Okay, sir. Thank you so much. And I'll maybe call you sometime later tonight or tomorrow, or maybe
Sure.
we can talk tomorrow because tomorrow is a
Sure.
public holiday. So I'll
Yeah.
give you.
Yes.
Right, sir. Okay. Thank you so much, sir.
Okay.
Thank
Okay.
you.
Okay.